Bismillahirrahmanirrahim. Today we are gonna study the spinal cord, but before going to the detail of it, we should know that our nervous system consists of central nervous system and the peripheral nervous system. And as you can see, the central nervous system consists of your brain and the elongated structure spinal cord. And you should know about the composition of your nervous system. We must know it contains neurons, the main cells, right, which are conducting, and the other would be neuroglia that are supporting specialized cell. And the third thing we have to mention, they must have intercellular substance consisting of blood vessels, interstitious fluid, and the ordinary connective tissue. But in CNS, which is brain and spinal cord, we expect very less amount of connective tissue, so it appears like a jelly, right? Now we have to concentrate the spinal cord, its features. See its extent. It starts from the lower border of the medulla oblongata, right? And ends up as a tapering structure called as conus medullaris. And the bony landmarks could be lower part of foramen magnum, right? And below your spinal cord ends in between the L1 and L2 lumbar vertebrae, in between its intervertebral disc, right? Now we know that our spinal cord being placed in your vertebral column is supposed to give a pathway for so many sensory inputs passing through it and reaching towards your brain, and from there motor inputs are supposed to come out and pass through it. For this you can see so many nerves coming out from the spinal cord. This spinal cord during embryological development is present at a higher level than that of your vertebral column. So your spinal cord ends up in between the L1 and L2 lumbar vertebra where the intervertebral discs lie. Now you can see that your spinal cord addresses two enlargements, one at the upper cervical level called as cervical enlargement, and if you have to tell the spinal segment, these are the C3 to T2 spinal segments, and the vertebral level must be C4 to T1 vertebra, right? And next to it, at the lower level, you can see the lumbar enlargement, which is inferior lying, and the spinal segment extends from L1 to S3. And if we quote the vertebral level here, we have to mention T9 to T12, right? Now, let me tell you the purpose of the cervical and lumbar enlargement. They are supposed to give rise to the nerves that comes in and go out, right? And there are so many, and the cervical enlargement is supposed to give the nerves to the upper limb, right? They have to arise from here. And the nerves of the lower limb has to get origin from that of your lumbar enlargement. That is why these areas are enlarged, and when we see the cut section, they show the maximum girth, right? Now again in this picture you can visualize the spinal cord that starts from the lower end of the medulla oblongata, then it addresses the cervical enlargement, right? Then you can observe the lumbar enlargement and can see the lower end of the spinal cord called as conus medullaris. Let me tell you that it ends in between L1 L2 lumbar vertebrae, and it is being said that in human body this level lies just a little above that of your elbow joint when the arms are hanging side by. And if you can observe at your abdomen, this region approximates to that of your transpyloric plane, right? Now we can see that from the conus medullaris, a prolongation of a fiber-like structure, the pia mater, is supposed to come down as a thin fibrous coat, right? Now in this picture again you can see the phylum terminale that is almost 20 centimeter long, and this above part is the phylum terminale internum that consists of 15 centimeter, right? And it extends or continues within the extension of dura mater, arachnoid matter. It has to reach up to the S2 sacral vertebra, right? And definitely here the dura and arachnoid matter, you would say, along with the subarachnoid space that are filled with the CSF, extends up to the S2 sacral vertebra. And then you can visualize the lower end of that phylum terminale, which is 5 centimeter long, is called as phylum terminale externum. Just 5 centimeter, right? It has to fuse with that of the investing dura mater, and it extends inferiorly after leaving the sacral hiatus. It joins to the periosteum of the first segment of the coccyx, right? And the central canal extends or continues into the phylum up to 5 to 6 mm, right? In this picture, you can see the transverse section of the spinal cord, and like the brain, you see spinal cord is invested by the three meninges. The outermost, which is shown in yellow color, is the dura mater. This is consisting of loose to dense connective tissue, fibrous strands, and attached to the periosteum of the vertebral canal. And below it there is a epidural space being filled with the loose adipose tissue and extensive venous plexus, right? And below that you find the purple color another meninge called as arachnoid matter, which is avascular, right? And now again below it you find the subarachnoid space, and then you can see the third meninge called pia mater that invests or appear as a plastered layer to that of the spinal cord. And the fact about the development of your vertebral column is that it lengthens to a greater degree than that of your enclosed spinal cord. So the spinal segments of the lower part of the cord does not correspond to the intervertebral foramen of the respected vertebral canal, right? So what happens that I would show you the nerves in the cervical region, they initially are doing the horizontal course, then they have to do the oblique course, right? Here you can see the spinal cord with its 31 pair of spinal nerves that runs in 33 vertebrae, right? Upper 8 cervical nerves, 12 thoracic, 5 lumbar, 5 sacral and 1 coccygeal, right? In the upper part, the nerves run the horizontal course almost, then they are obtaining the oblique course till you reach the lower level of the spinal cord. Here lower spinal nerves, they are forming angle, and in the vertebral canal they appears like that of a collection or wisps of hair, you would say, and they are here known as cauda equina, which means like a horse tail, right? In this picture again we are looking into the spinal cord and the spinal nerves, and there is an enlarged view of the vertebra and the transverse section of spinal cord that is showing what? Here you concentrate the positioning of it. The region of the spinal cord associated with the emergence of a pair of nerves are called here as a spinal segment. You should know that when you see the transverse section of the spinal cord along with the spinal nerves both on side as forming a pair, so a spinal cord and the nerve. They both are under the heading of spinal segment, right? You should. Here in this picture, you can see the histology of the spinal cord. The transverse section of it is showing its oval diameter, you would say, and it is flattened more anterior than posterior, right? And in this picture, what else you can see? The outermost covering, which is darker in stain, you can find it as dura mater, right? Next to it, the middle layer is the arachnoid matter, and you can find the third, that is more clung to your spinal cord, is the pia mater, and on its very margins you can observe various cut section of the blood vessels, right? And now you can concentrate on the various fissures and sulci. Anteriorly, there is a deeper fissure called as anterior median fissure, being marked on its anterior aspect by the pink arrow, right? And on the posterior aspect, you can see the shallower sulcus called as posterior median sulcus, right? Being again marked with the pink line, pink arrow. And on the lateral aspect anteriorly, you can find the anterolateral sulcus being marked with a light blue color, and on posterior aspect you can find the dorsolateral sulcus marked with the purple arrow, right? Here in this picture, you can visualize the central located darker stained area called as gray matter, which is an H-shaped structure or like a butterfly, right? And let me mention you about its various horns. Let me mark with the arrow that the most prominent anteriorly placed are the ventral horns, and the smaller one are the posterior horns, right? And you can observe the central canal is present in between it, and that is being filled with the CSF, right? And the part of this gray matter that joins anteriorly in the middle, anterior to this central canal, is the anterior gray commissure, and the part of the gray matter that joins this H-shaped structure in the middle behind the canal is called as posterior gray commissure, right? Here in this picture you can see the four major region cutted as a transverse section of the spinal cord, namely the cervical, thoracic, lumbar and sacral. And we should know that the structure of the spinal cord basically remains same in its whole length. But what happens? There must be some functional differences among these four major levels. Now we will discuss them one by one. You can see the transverse section. Now in this, now what is going to be shown? The ventral horn are more prominent because they consist of the cell bodies of the large lower motor neurons to supply your muscles, and the dorsal horns, which are smaller and much less prominent. What they carry? They carry the cell bodies of the second order neurons, which are sensory in nature, and they have to relay sensory information coming from your receptors present on the body to receive temperature and pain, and their cell bodies lies, if you can recall, the dorsal root ganglia on the spinal nerve, right? The important thing in these transverse section, you can see that the gray matter volume is much more extensive in the upper cervical region and especially the lumbar region, because they have to supply the great sensory and motor innervation for your limbs, and that can be reflected due to its greater diameter of the spinal cord here, right? And what else could be there? That the ventrolateral white matter is again addressing, and is supposed to give pathway for the ascending and descending track here, right? That is why this cervical region is showing more girth. Let me tell you some other details. We can learn that if you want to see the differences of these different areas, that why their diameter or girth is different in different regions, so it must be understood that the ascending tracks, if you keep in mind, the number of fibers is keep on adding in each ascending segments, right? So the segments will be addressing more number of fibers which are increasing up to they have to reach the higher levels from sacral to the cervical above. And same happens with the descending tracks. What is going to happen? That if you concentrate the top cervical segment, here the coming descending tracks, their number of fibers now keep on diminishing at the lower level. Means that the number of fibers should be terminated downwards at each cervical to thoracic to lumbar to sacral segments. Their number is decreasing. So we expect that the quantity of white matter must be much more in cervical segment levels, and they are keep on decreasing in descending orders in that of the thoracic, lumbar and sacral levels, right? And if you concentrate similarly, the quantity of gray matter is much more in your ventral horn at cervicothoracic and lumbosacral region, right? If an it could be marked like this, if you can concentrate, because they have to address the cervical and lumbar enlargement to supply your upper and lower limbs. So we expect that the upper cervical, thoracic and lower sacral segment are showing now the lesser quantity of gray matter, but the middle cervical and the lumbar region are addressing the more gray matter, right? And now another important thing about the location of the central canal. See, in the cervical region and in the thoracic region, here what is going to happen? It is located more posteriorly because increased number of fibers in the dorsal column track of higher level, right? So your canal appears posteriorly, but it lies more anteriorly if you concentrate here, that in the lumbar and the sacral region it is placed anteriorly, right? What else is there? Now we have to see that the central canal, you can visualize, is being aligned with the ependymal cells. Let me show you another picture for it. These are the ependymal cells that lines the central canal and as well as the ventricles of the brain. So in the spinal cord you can see these ependymal cells having no basal lamina, but these cells are either columnar ciliated or cuboidal ciliated, right? Now we have to learn in general that the gray matter, which is a seat of intelligence in spinal cord, you would say, it consists of neurons or neuronal cell bodies and their dendrites, right? And we expect the initial unmyelinated axons be a part of a gray matter. And the last thing are the neuroglial cells. These are the structures you would observe in gray matter of your spinal cord, right? And if you concentrate the white matter of your spinal cord, that would be showing the myelinated axons mainly, but some unmyelinated axons could be a part of it. And you expect the neuroglial cells which are supporting in nature, for example, oligodendrocytes, right? This is again a very good picture to show you the placement of neurons. You can visualize, and along with its cell body and dendrites and axons and other supporting cells, which are, you can see, astrocytes, and you can observe the oligodendrocytes and some myoglial cells you can observe, right? These are the contents you can find in the gray matter of your spinal cord, and the lower lining are the ependymal cells. You can see in marked in a yellow color, which are simple cuboidal or columnar ciliated, right? Now in this histological section, you are looking into your screen. The left side of your screen is showing the white matter, and right area is showing the gray matter. Gray matter would be observing the neuron or the neuronal cell bodies, and there could be the glial cells, which is marked with the letter G, right, all around. And the white matter is addressing so many axons all around, right, and the glial cells. But here neurons are not a part of it, right? In this picture again, which is a gray matter, showing the neurons. Neurons, here you can see the neuronal cell body, and this whitish central structure in the neuron is its nucleus, and in the very center there is a very well marked nucleolus that is lying, and the cytoplasm of the neuron is filled with the Nissl granules or Nissl substance. These purple color marking you can observe. What are these? These are basically the rough endoplasmic reticulum along with the free ribosomes, and they are showing that they are supposed to produce the structural and transport proteins, right? Then you can see an area which is an axon helix that is not showing the this Nissl substance in it, right? And here again you can observe some blood vessels, that is an arteriolar structure being cut, marked with the arrow, carrying blood cells in it. And these dotted darker colors structure, these one are the helping supporting glial cells, and that reddish spot could be the capillary present in the gray matter, right? Rest of the pinkish stained areas are showing various axons and neurofibrils in between, right? This is again a very good diagram of the spinal cord transverse section. Center structure is the gray matter, and in the very center, central canal is lined with the ependymal cells, right? And the gray matter you can see is having the glial cells in it, and the anterior horn is addressing the motor neurons, which are multipolar or Golgi type 1, right? And what else you can see on the margins in the white matter, all around the myelinated axons are being there. Some capillaries could be a part of it, and surely the glial cells are present in the white matter as well, and they are supposed to produce the myelin sheath, and when we stain it out, sometimes these lipid content of that myelin sheath are washed away and they appear empty looking sheets around the axons, right? And the darker stained are the glial cells, oligodendrocytes in your white matter, right? Yeah, in this cut section you can observe various neurons, interneurons, right? You can see that the ventral horn is showing the cell bodies of the motor neurons, right? And the posterior horn is addressing the processes or the neurons, sensory neurons, which cell bodies are definitely located in the dorsal root ganglia of the spinal nerve, right? And how these neurons are interconnected, these are through various interneurons. And let me tell you that this motor neurons and most of the interneurons, they are almost 99% made up of multipolar neurons, right? And you can observe that there is a muscle present which is skeletal. You can call it as effector muscles. Here synapses occur through the synaptic knob. The information is taken from the receptor and ascending upwards as a first order neuron or first in command order they have to reach to the sensory neuron cell body present in dorsal root ganglia. From here, afferent sensory information is moving through the dorsal root into the posterior gray column, and then the interneuron is shifting that information towards the other side of the limb of the gray matter, and you can observe the cell body of the motor neuron in the ventral horn. That is a map you should learn, how the information reaches towards your brain and moving upwards and downwards. Here is a very important diagram to learn about the neurons, how they reach towards destination. Concentrate that first box is showing the receptor. From here, first order neurons, they take information towards your spinal cord and reaches to the relay nucleus. They are first in command. From here, second order neurons, they ascend upward through the brainstem and reaches your thalamus. And from here, after this 2IC or second in command, second order neuron, in the thalamus now, the third order neurons, they are supposed to take information towards your cerebral cortex, and from here towards the respected area, the fourth order neuron is taking information towards the required site of the cerebral cortex, right? Another important diagram I have taken for you, that the information like fine touch, proprioception and vibration. And other column is showing nociception, temperature and coarse touch. They are moving from the receptor and up to the dorsal root ganglia. From here, cell bodies of the sensory neurons lie. The information are entering into the dorsal horn of the gray matter of the spinal cord. These first order neurons, they are ascending upwards, now reaching towards the medulla oblongata. From here, second order neurons are supposed to reach towards the thalamus, and the third order neurons are supposed to enter via that cerebral cortex, and the fourth number is showing the final destination of this whole track. The blue color are showing the first order neuron, and the red color marking are for the second order neuron, and the black color is showing the third order neurons, right? On all the left side. Right side, again you can visualize the pathway, and in the very lower first column you can see that how the dorsal horn, from here information of this neuron is moving through interneuron using the ventral and dorsal gray commissure, reaching towards the other limb of the gray matter, and then ascending upward up to the thalamus, right? And information we have already mentioned that the pain, temperature and coarse touch now have crossed the midline in the spinal cord, and which is very clear in this picture. And then you can see the fine touch, vibration, proprioception pathways cross a midline in the medulla oblongata, right? This area is being shown here. And thirdly, sensory pathway synapse in the thalamus. And fourth one, sensations are now perfectly reached and perceived in the primary somatic sensory cortex. So that map helps you a lot about the ascending tracks, how they work, right? Another important thing here you can observe in the longitudinal view of the spinal cord, that the pia mater is being shown in yellow color, and on its margin the pink color area is addressing almost 21 pairs on the lateral aspect of the spinal cord, which are triangular extensions of the pia mater, and they resemble the shark's teeth. So they are called as denticulate ligaments or ligamentum denticulatum. They are supposed to pass through the arachnoid to reach to the dura mater on both sides. So we should learn this thing also, right? Now the very important thing to be discussed here are the intermediate horn or lateral horn of gray matter in the thoracic region, I would say, and it is also present in your upper lumbar region, right? And what they carry? They carry inside the cell bodies of the preganglionic sympathetic efferent neurons, right? And they are supposed to be concerned with your sympathetic outflow from the cord, right? This diagram will be showing you the transverse section of the spinal cord, and you can see that how spinal nerve is taking sensory information which is marked by a red line and reaching towards your dorsal gray column, and from here the axons which are entering into the white matter either on the lateral aspect or on the dorsal white column, and they are ascending upwards. This is the pathway to show you various tracks, right? Now this is the slide I want to show you, which you will observe in your lab, is this spinal cord. The darker central region is the gray matter and the lighter region is the white matter, and you can observe the anterior median fissure, the posterior median sulcus, the dorsal white column, the anterolateral white column showing all the axon in it, and the central canal is there along with the anterior and posterior commissure, gray commissure I would say, and the gray matter as a butterfly shaped structure carrying the neurons, the glial cells, and few the axons and dendrites be a part of it, right? In this schematic representation, if you can see the neuron, its cell body, and you can find so many branching processes called dendrite, and below you can find the axon, and the very initial part through which it starts is called as axon hillock, and this axon hillock is not showing the Nyssel's bodies, rather they are present in the perikaryon or the cell body of the neuron, and also in the dendrites as well. And you can see that the oligodendrocyte which is present nearby as a supporting glial cell is forming the myelin sheath around the axon. So keep in mind all these details. In the name of Allah, the very important topic to be discussed right now is the blood-brain barrier. What it is? It is a highly selective functional barrier present in your CNS to prevent the passage of various toxic drugs, the antibiotics, and some bacterial toxins, because they have got some harmful effects. So if substance get entry to your bloodstream, they shouldn't pass towards your neuronal tissue of the CNS. And if electron studies are to be done, we should know that it depends upon the tight junctions present called as zonuli occludentes, right? And they are present in between what? In between the endothelial cells of your blood vessels, especially the blood capillaries of the neuronal tissue of the CNS, right? And the other thing that come across are the astrocytes. Through there, if you can concentrate in the picture, the blue color are the food processes of these astrocytes. They, along with the endothelial cells of the blood capillaries, they mark your blood-brain barrier, and they almost completely surround the basal lamina of these endothelial cells, so can play a very important role in maintaining these occluding junctions present in between the endothelial cells of these continuous capillaries of the brain and the spinal cord, right? And if electron microscopic studies are there, they show very few pycnocytotic, we would say, vesicles in the cytoplasm of these endothelial cells of your brain capillaries, because the substances that do cross the endothelial cells must be transported by receptor mediated endocytosis like oxygen, carbon dioxide, water, and some small lipid soluble material including few drugs easily pass through the endothelial cells, right, of your brain capillaries. And the glucose, amino acids, vitamins, and nucleosides are transported through a process called actively transported process across your endothelial cells by specific transmembrane carrier protein. So we need this blood-brain barrier intact for the healthy functioning of